Hi class, Mrs. Klein's here with your first section of video notes from our Unit 1 in Zoology, Phylum Porifera. Now we're going to start by talking about some porifera basics. So let's first dive into the name meaning. Porifera means pore-bearing or covered in pores, and those pores are called ostia. The Latin term ostia means doors. The pores are the doors into the sponge. Currently, the number of living species in phylum Porifera is around 8,550. This number is constantly changing. There are many deep sea sponges that we have recently just discovered. A couple other Porifera basics. The examples from phylum Porifera are going to include sponges. Their symmetry is asymmetry or asymmetrical. Asymmetry indicates that this creature is sessile, so that means fixed in one place or immobile. And then classifying them as a triploblast or a diploblast, we would call them diploblast. With being diploblastic, this will greatly impact their body plane. Sponges are basically two layers of cells. So they have an ectoderm, they have an endoderm, and that is filled with a soft or hard skeleton. The next classification we'll talk through is protostome or deuterostome. And for porifera, we actually classify them as neither of these. Sponges lack both a mouth and an anus. So while they lack a mouth and an anus, they do have an entrance and an exit to their body plane. The ostia or the pores are the entrance. The osculum, or the large opening at the top, like you can see in this image here, is the exit. Habitat is the next thing we'll talk about. Most sponges are marine, so they live in oceans or seas. And just a quick recap, an ocean is larger than a sea and is not contained by land. A sea is an extension of the ocean that is partially enclosed by land. And they can live at varying depths of water, so they could live up to 5.5 miles deep. Continuing with their habitat, there are roughly 200 species that live in freshwater, and sponges cannot survive on land. So there are 31 species currently identified in North America. The freshwater sponge featured on this slide is a Spongilla lacustris. Lacustris is a Latin word that just means lakes. So this one is one that is found in North America. Next, we'll talk about some unique characteristics. So sponges have been around a long, long time. We have some fossils that we have found from over 500 million years ago. They were one of the first animals on Earth. So there was a fossil recently discovered that is suspected to be a fossil of a sponge from phyla Porifera. It is over 890 million years old, and it is highly debated whether sponges or comb jellies were the first animals on Earth. Current evidence points more towards comb jellies being the first. A couple other characteristics. Unlike other animals, sponges lack tissues and organs. So quick refresher, remember a tissue is a group of cells that work together with similar anatomy that perform a similar function. An organ is a group of tissues. Sponges do have cells and specialized cells at that, but they do not form tissues like blood, bone, cartilage, fat, etc. We also say that all sponges are sessile. So as I mentioned earlier, they are sessile, meaning they're immobile. But the amoebocytes, which we will talk about a little later on, enable some sponge species to be able to move along the ocean floor slowly. All right, if you look, quick question, what do you think you call a group of sponges? Are they called a shiver, a colony, a caravan, or a muster? If you guessed colony, you are correct. A group of sponges living together is known as a colony. Okay, couple more unique characteristics we'll talk about. Most sponges are filter feeders, so none can do photosynthesis. Some of them can hunt, but most are filter feeders. And then sponges are often brightly colored, as seen on this slide, and they do produce toxins. These toxins deter predators and competition. We have also begun, as humans, using these toxins in certain medicine. So these medicines will include drugs to help treat herpes, HIV, and even cancer. Okay, and this is where we will pause for your first round of video notes. So make sure you followed along and are caught up on your note guide.